Welcome to the Brownback Lecture Series from BRIT, the Botanical Research Institute of Texas. This BRIT Brownback Lecture Series takes place the first Tuesday of every month. I'm Alejandra Vasco. I'm a research botanist here at BRIT. And here in the studio with me are Dr. Weston Testo, today's speaker, and who I will introduce in a minute, and Dr. Manuela Dal Forno, also a research botanist here at BRIT, who I thought will be only helping me with the chat today, but she did everything and entertained people in the other session we had deleted for some reasons. So thanks so much you both for being here and for sticking here. Before we started, I would like to mention that this lecture and the accompanying chat on YouTube are both being recorded and will be available in the future on the BRIT YouTube channel. So if anybody misses the talk because of the wrong link, they will be there. Also, I want to remind you and encourage you and our viewers to ask questions using the YouTube chat during the whole lecture. And at the end of the lecture, our speaker, Wes, will try to answer your questions. Also, if you can tell us where you are, where in the chat where you are watching us from, that would be great. So I think these are all the announcements, and now I can finally introduce our dear speaker, Dr. Weston Testo. Wes is a research associate here at BRIT, and he's currently a postdoc in the Antonelli lab at the University of Gothenburg in Sweden. Wes has a PhD in plant biology from the University of Vermont and did his undergrad at Colgate University in Hamilton, New York. During his whole career, Wes has been interested in different aspects of the evolution of ferns and lycophytes. He's an avid collector and supporter of collection-based research, which I'm very happy about. He has also been especially interested in understanding lycophyte diversity in the American tropics, and he's been also interested in the systematics and taxonomy of the clubmoss family, Lycopodiaceae. So today, Wes will be talking to us about the commonness of rarity, evolution, and endemism in South American clubmosses. And before I let you talk, I want to say that Wes has done many things, and it's very difficult to introduce him in 2 minutes. And after all this crazy, so I will encourage you to look and to visit his website to learn more about the amazing things he does. And on a more personal note, I would like to say that Wes is a great colleague, a great field workmate, a passionate teacher, and an avid plant collector, and above all, a dear friend. So thanks so much, Wes, for being here and for sticking out with us. Now, I will give you the stage. Thanks.
Okay. Thank you so much, Alea. Everyone can hear me okay? Is that good? Well, thank you very much for that wonderful introduction. I hope everyone can see my screen. I appreciate everyone sticking with us through that little bit of technical difficulties, but if that is the worst thing that happened to you in 2020, you're having a pretty good year. I think what actually happened was I was supposed to be streaming on the national news, but I guess something's going on today in the US, and I got boxed out. But today I'm going to be talking about rarity and evolution of club mosses in the Americas, with a focus on the genus Phlegmariurus, which is a group that I've worked on a lot during my PhD and since. But I want to start off kind of broadly talking about rarity and what it means for biodiversity. I think it's important to keep in mind that, although some species, like the ones that you see on the left, are weeds somewhat, that is not the case for most species on Earth, right? So humans, Japanese beetles are kind of weedy, but there are many, many things that are rare, and that's a pattern that we see repeated across the world. There's many reasons why species can become rare. There's a lot of really interesting literature on this, and I'm just going to touch on it really briefly, some kind of common themes. Species can become very rare if they're extremely specialized, right? So this red cockaded woodpecker is a really great example. It's a bird that relies on a very specific ecosystem. That ecosystem becomes rare. It's kind of stuck between a rock and a hard place, so to speak, right? There are species that are rare just because they occur in a place that's very isolated and very small, right? If you only live on a very small island in the middle of the ocean, you're probably never going to be very abundant no matter what. There's, of course, species that are kind of in long-term on decline, so to speak, across deep evolutionary time. Right? So something like the tuatara was once more abundant than it is now. It's been around for many, many millions of years, and now is just restricted to a relatively small part of Earth's surface. And of course, increasingly, pressures by humans obviously cause rarity and extinction in many taxa. And I don't think I really need to spend too much time on this, but it's important to keep in mind that, rarity does matter, of course. Patterns of global biodiversity are in large part shaped by rarity. Like I mentioned before, there's some species are very widespread and common, but the majority of species are actually rare. So if we think about places with a lot of diversity of different taxa, a lot of that diversity is comprised of species that are rare and often endemic, right? And endemic is a bit of a relative term, but in case people aren't familiar with that, it's just this concept of a species that only occurs in a given area. Of course, species that are rare are more likely to go extinct, right? You can imagine that if something's only known from a couple of small populations, it's quite likely that it may go extinct in the near future. And of course, we're seeing around the world that overall more and more species are becoming rare and going extinct. And this is important to keep in mind because from a management perspective, of course, conservation resources are finite, right? There's only so much money, time, and effort we can put into conserving species. So having some understanding of what causes things to be rare and which taxa are rare is really important. And for many groups, we simply do not have that sort of information. So if you think of this globally, it's quite interesting that rarity is not evenly distributed. Like I mentioned briefly in the last slide, hotspots of biodiversity are also hotspots of rarity. This is a really wonderful paper. It came out last year. Other papers have explored this theme, but I thought this one was real nice, led by Brian Enquist and many other collaborators. If you look at this map, the kind of darker colors, the darker kind of oranges and browns, indicate places where there are a lot of rare species. And you see that those places overlap quite nicely with places that we tend to think of as places of really high biodiversity, right? So tropical America, South Africa, Madagascar, the islands off of Southeast Asia and Eastern Asia, all have a ton of rare species. And if we put that in the context of thinking about change moving forward, we obviously live in a rapidly changing world. The future of rarity really reflects patterns of biodiversity in much the same way we have here. So this is from the same paper. And basically, the hotter colors that you see here are areas that are predicted to have a greater kind of increase in rarity, if you will, so that kind of biodiversity crises will get worse in these areas relative to others. We see that this overlaps with where we already have rare species. So it's only going to get worse for those places in the future. And of course, thinking about this across the tree of life, it is perhaps not surprising to think that not all groups of species are equally rare. There's certainly certain clades, certain groups of lineages that have a lot more rare species than others. One that comes to mind for a lot of people is orchids, right? There's a really nice paper by Alex Siska and some other folks just this year that pointed out that an incredible percentage of orchids globally are rare, right? Certainly, I think some of you can probably think about some other groups of animals where a lot of the species in that particular group are currently rare or in decline. And I think quite often a common thing that we think about when we talk about rare species is that they're kind of old lineages kind of on their last leg, right? I already mentioned the tuatara, right? This is a lineage, one species currently has been around for about 250 million years. The ginkgo, again, one species, almost 300 million years. The coelacanths, right? Something that's been around since, the Devonian. There's just two species. And scientists, didn't even know they were still around until relatively recently. But kind of conversely, if you flip that on its head, there's a really interesting pattern that in reality, most rare species in the world actually belong to very diverse groups. So I pointed out just 3 very species-rich families of flowering plants, all of which have a lot of threatened and endangered species, right? So the Fabaceae, the pea family, almost 20,000 species, about 1/3 of them are threatened or endangered based off of our understanding of species that we have examined for rarity. The coffee family, Rubiaceae, almost half of the 19,000 there are considered threatened or endangered. And in the sunflower family, it's over half. So there's kind of an odd coupling there. It seems a little bit odd that an extremely diverse group of organisms would have a lot of rare species, but that is often the case. And piling on top of that and kind of tying these themes together, the reality is, as we've seen in that earlier map, that most rare species in the world belong in tropical places. Now, there's a lot of reasons why this might be. Some of it has to do with the fact that species in the tropics tend to have smaller ranges. So again, I'm here in southern Sweden currently, and if a birch tree growing here were to survive throughout the year, it has to deal with really high variation in temperature, right? It certainly gets below freezing, and then temperatures get relatively warm in the summertime. However, if you're in the tropics, in most areas in the tropics, there's low seasonal variation in temperature. So you can imagine that species could kind of have a very limited niche with respect to the temperatures that they need to tolerate to survive. And that can ultimately result in many species having very small ranges, especially along gradients of perhaps precipitation, but especially along mountains. If anyone's ever gone to the tropics and driven along a mountain road there, you know that it goes from very hot at low elevations to quite cold all year round at high elevations. And so you switch through a lot of different habitat types in the tropics as you go up elevational gradients, more so than you would if you went across the same gradient in the temperate region. So that's one thing that contributes to it. Another kind of interesting component is the idea that some tropical areas, through kind of deep time over the course of hundreds of thousands or millions of years, have relatively stable climates. Certainly for viewers in Europe or in North America, you're probably aware that there's a significant history of glaciation, especially over the last few 10,000s of years, that impacted the biota of those areas immensely. Certainly there's a lot of climatic shifts in the tropics too, but not kind of this wiping clean of the slate. And so there seems to be lower extinction in some tropical areas, and those kind of function as biodiversity museums, if you will, which might allow rare species to persist longer than they would in some other places. So to kind of think about what makes something rare and what drives rarity globally, I think your perfect study group in some sense has to kind of link all of these different components. So you have kind of an ancient group would be really interesting. Particularly things in the tropics seem to be quite interesting. And this interesting pattern that I mentioned before, that many of these groups of organisms that have lots of rare species tend to be species rich. And one group that I think does this pretty well are the lycophytes, the clubmosses. So lycophytes, I have a phylogeny here, an evolutionary tree of land plants. Lycophytes are a very old lineage of plants, over 430 million years old, and they are sister to the rest of the vascular plants. So for people who aren't familiar with that, the idea is that they diverged at the same time that the rest of the vascular plants did, and that they're the closest related lineage to ferns, gymnosperms, and angiosperms. It was once a dominant group, right? If you go back to the Carboniferous, this drawing kind of depicts that here. These plants dominated Earth's landscape, but now not so much. If people are familiar with club mosses, it's often that they see kind of puny plants growing in bogs or in relatively nutrient-poor soils quite often. But thinking about it globally, lycophytes have some really diverse groups, but they have a lot of rarity. It's a really fascinating group of plants in which many of the species are only known from a few places. So here's some of that fossil diversity here. You can get a sense of how varied these life forms were hundreds of millions of years ago. And they're quite variable today too. I mean, certainly they're not forming trees that are 20 meters tall, but there are aquatic plants like the Isoetes in the bottom middle there. There's plants that grow hanging off of trees, like the plant in the top middle, ones that grow in really dry habitats, ones that grow near the Arctics, and ones that grow in the tropics. So it's a really fascinating group in a lot of ways. Modern lycophyte diversity is broken down into 3 families and about 1,200 species today. So the 3 families are the Lycopodiaceae, Selaginelliaceae, and Isoetaceae. The Selaginelliaceae and Isoetaceae are each other's closest relatives. They're heterosparous, which means they make 2 different kinds of spores, whereas the Lycopodiaceae are homosparous, make 1 kind of spore. Lycophytes have a worldwide distribution, but they're most diverse in the tropics, just like we saw for many of these lineages with many rare species. The kind of morphological characteristics that define them are that they have microphyls, which are leaves that are supplied by just a single vein, so they're very small. They're spore dispersed plants. They don't make flowers, they don't make fruits. And the sporangia, which hold the spores, are on the adaxial surface of the leaves. So that's to say, kind of the top side of the leaf. You can see that in the image at the top, right? Those kind of clam-shaped structures, those are the sporangia. They sit on the top side of each leaf. So that's what makes a lycophyte. And today I'm going to be talking about this family that I've circled in red here, which is the Lycopodiaceae. Lycopodiaceae, I think, is a really interesting group because in some senses, it looks like a little bit of a renewed radiation. Again, I mentioned that lycophytes were once very diverse, not so much anymore, but the Lycopodiaceae includes 400 to 500 extant species that we've documented so far. It's a family with a worldwide distribution. So you see that one is hanging from a tree. That's from Colombia. That one amongst all those beautiful cladonias there, those lichens, is a species that's very common in the Arctic. And then there's this giant one that's kind of hanging off the trees like a vine. So they're all over the place. As you can see, they're ecologically diverse. There's some evidence that maybe there's recent diversification in the history of this family. And there's many rare species. Now, to do any sort of analysis of rarity or evolution of a group, you need a good timeline of the evolution of the history of this group. And when I started my PhD, really all we had to work off of was a single phylogeny, a single evolutionary study that involved 1 gene and 33 species that was published in 2001. For the time when it was published, 2001, incredibly advanced study and really changed a lot about what we thought about the evolution of this group. But for the sorts of analyses that we need to get a really in-depth understanding of the evolution of plants, just was not sufficient. So we really needed an improved framework pretty badly. I'm not going to get into this too much because it's not the main point of today's talk. But one thing that I did do over the course of my PhD was kind of generate a timeline of club moss evolution. So I went and got samples from 200 species. So that's about 50% of the global diversity in the family. It involved fieldwork in 11 countries, mostly in tropical America, but in some other places as well. Also going to 43 herbarium and working in the collections there to fill in gaps. Because obviously natural history collections are so important to much of the research that we do. And then using the program Beast version 2.4, integrated data from 6 chloroplast DNA loci, 8 fossil calibrations. Obviously, there's lots of fossils of lycophytes, although it's really hard to pin them down to a specific family in many cases. And they used what's called a random local clock model to associate rates of nucleotide change through time with evolutionary time, right? So the kind of basic premise of how these analyses work is you have a DNA alignment, you estimate changes in DNA, and those give you the kind of branch lengths in your tree. And then you need to convert those branch lengths from substitutions of DNA to time, and there's various ways to do that. And the random local clock model allowed us to do that in an effective way in this family because unlike many other groups of organisms, we had a lot of evidence that evolutionary rates in the Lycopodiaceae and its close relatives varied a lot. And so it was very difficult to kind of transform branch lengths from nucleotide substitutions to evolutionary time. Again, it's not the main point of what I want to talk about today, so I'm going to kind of gloss over it a bit. If anyone has questions about that, I'd be very glad to have a more in-depth conversation. But what we ended up with was a pretty nice timeline. So I'll just point out real quick, we have this evolutionary tree of lycophytes, and it is a group that goes back about 400 million years. So in the red colors are the Lycopodiaceae and the different clades, the Lycopodiaceae in the bright red and in the darker red. And in blue are the heterosporous lycophytes, the Isobutaceae and Selaginellacea. So we see that the deepest divergence here is between the Isobutaceae and Selaginellacea and the Lycopodiaceae about 400 million years ago. And then we see divergences within the Lycopodiaceae starting at about 360 million years ago. So it's a really ancient family here. So we're talking about families of plants that diverged in the Devonian. And this is really incredible. And the biogeography, thinking about the distributions of different lineages within the family, those really correspond to the breakup of supercontinents through time, which is really rarely found in other groups of organisms. So this is a case where, Pangea started to break up probably about 180 million years ago. That corresponds to some distributions that we see in many of the genera of the Lycopodiaceae today. And then later, Gondwana, which was kind of continents that we see in the southern hemisphere today, that started to break apart into South America, Africa, Australia, and a couple of different components. That happened about 100 million years ago, the last kind of rifting there. And that corresponds to splits that we see in groups between the Americas and Africa and Asia. So that's a really kind of amazing signal of the past in this group that you really don't see in many other groups of organisms. And then in these kind of bright red branches, inside that dashed box, you see a lot of really short branches indicating very recent evolution. And that is rapid diversification in the genus Phlegmariurus. It's a mouthful, but that's going to be what I'm going to talk about for much of the rest of this talk. Phlegmariurus, the tropical fir mosses, we often call them, these plants were historically often treated as part of the genus Huperzia, but we now know that true Huperzia is actually mostly restricted to temperate parts of the world. This is the clade with about 300 species worldwide, so that's 80% of all the species in the entire family. It has a pan-tropical distribution, but as I mentioned before, it has these paleotropical and neotropical clades that correspond to the kind of final breakup of Gondwana about 100 million years ago. This group is most diverse actually in South America, especially in the tropical Andes. I'm going to talk about that a lot. And when you get to the tropical Andes, you see a lot of rarity in this genus. So Phlegmariurus in the Neotropics includes about 150 species. These are ecologically very diverse plants. You gotta remember, these plants are just branched stems with little tiny kind of fake leaves hanging off them. And despite being that simple, they really are quite successful in doing a lot of different things. And there's basically two big groups that you see. One are species that are epiphytes, so they grow hanging off of trees. Those are mostly in cloud forests. They can be found from sea level up, but are most common between about 1,500 meters elevation up to tree line in the tropics, which is usually a little bit over 3,000 meters. And then there's a big group of terrestrial species, which we find in alpine habitats, many of which are called paramos, especially in the northern Andes, or Puna and other parts of the Andes. And those are generally found above 3,000 meters elevation, and I've collected them up to about 5,000 meters, which is about where you start to hit snow. It's extremely species rich in the Andes. This map here shows collections of Phlegmariurus in the Americas, and you can see that there's just a ton of them in Central America, southeastern Brazil, and especially in the tropical Andes. And many of these species appear to be rare from everything we can find out about them. And this is what they look like. Again, it's a really diverse clade in terms of the number of species, but also how they look. And you can see these two kind of main variations on a theme that we see over and over again, right? So there are epiphytic ones, or in the case of that one on the top, apparently growing off of someone's house. And then there's these terrestrial ones, which are often kind of bright red, which seems to be an adaptation to growing in habitats with really intense sun. And I have clicked back, excuse me, I clicked over there, one extra slide. And when we look at Phlegmariurus across the American tropics, we find out that there's really interesting patterns of diversity, and it's not even throughout the American tropics. So I've done field work in a lot of places on this map, and it is much easier to find Phlegmariurus species in some places than others. So Phlegmariurus, again, occurs from Florida in the USA and Mexico all the way down to Bolivia, Argentina, Brazil. But if you go to northern Central America, let's say you go to Nicaragua on the map there, you'll find about 15 species in those countries. If you go to the Amazon, you'll only find 10 species. Lots of people talk about how diverse the Amazon is, and it is for many groups. There's only 10 species of Phlegmariurus there. If you go to the northern Andes, if you go to Colombia, or you go to Ecuador, or Peru, or Venezuela, you can find as many as 100 species in a country. So what's causing that? Well, I think a lot of it comes down to topography, right? So this figure I've kind of put here has on the y-axis elevation. And what it is essentially a profile of the landscape if you've kind of walked from that one red line to the other, right? And so you see there are some mountains in northern Nicaragua. You get up to about maybe 1,500 meters, maybe 4,500 feet or something like that. And again, those in the kind of top left of that figure, there are, I think, 15 tiny club mosses, so you can get a sense of how many species are there. So the mountains aren't that high in Nicaragua. And if you go to the Amazon, as you might guess, looking at this red line, it's even lower, right? It's pretty much flat. And there's even fewer species. But if you go to the Andes, that blue line, the mountains are big. Right, these are really dramatic mountain ranges, and they allow for a lot of species to occur. Again, around 100 species that occur in this area. And when you look at Phlegmariurus in the Andes, you see species sort out in this really interesting way, which I already alluded to earlier. You get epiphytic species at these kind of middle elevations in the forest, habitats that look like that. And then you get these terrestrial species at really high elevations. And that was a really interesting thing to me when I started studying this during my PhD work. Now, certainly we know from a lot of other groups of organisms and from geological data that the Andes are a biodiversity hotspot and that they have lifted up, especially in the northern parts of the Andes, relatively recently. So from a geological perspective, the Andes are extremely young. And the northern Andes approached their modern heights probably in the last 5 million years. So when you have mountains that tall in the tropics, the tops of the mountains are so high that trees can't grow there. So you have alpine zones on tops of the mountains, which effectively are like islands in the sky, right? So you can see what in the background of this image, there's no trees there. But if you go down off the edge of that slope, there's rainforest for a very long time. And so species that live on these kind of alpine habitat islands are isolated. There's limited gene flow between these, and we know that through time, changes in climate drive pretty dramatic shifts in how big these alpine habitats are. From an evolutionary perspective, this is really interesting because it provides this kind of novel opportunity for species living in the forest below to explore new niches as these habitats arose, right? So as the Andes pushed up and got bigger and bigger and bigger, and they started to get so big that trees couldn't grow at the top, that was a really interesting opportunity that some species could exploit. And we know from other groups that there's lots of, especially flowering plants that have done very well, diversifying very rapidly in these habitats. So these are all flowering plants again. On the left, the kind of small plant, that is a gentian There is Puya raymondia, this very large member of the pineapple family there. Lupins, perhaps a plant that people know from gardens. Those are extremely diverse in the American tropics at high elevation. And then these kind of peculiar looking little tree-like plants are actually members of the daisy family, which have diversified extremely rapidly in high elevations in the Andes. So what about Phlegmariurus? Why are there so many species of this group just in the Andes? Is it the same kind of pattern that we see in a lot of flowering plant groups? Basically, I had three questions, kind of trying to understand the evolution of the group. And I will get back to the rarity stuff in a little bit, but the three big questions were, is diversification in this group linked to the uplift of the Andes? If that was the case, you would see increased rates of diversification in young alpine lineages compared to their forest-dwelling relatives. I wanted to know, when did terrestrial growth evolve? This is an extremely old group. Is it possible that the terrestrial species just migrated from temperate regions where there's already terrestrial species in other clades? Or was this a very recent diversification in these alpine habitats? If that was the case, we'd expect this recent transition from growing as an epiphyte to somehow growing on the ground. And do we have any evidence of this kind of sky island pattern of speciation, where we would expect to see species at high elevations having very narrow ranges and probably being very rare compared to species growing in the forest below? So this is a really complicated past, and I just want to touch on the methods that I use to explore this a bit. So I took that timeline, that phylogeny that I already showed, pruned that down to just include American species of Phlegmariurus. So that included 106 species, about 70% of the species in the genus. Pulled out distribution data and ecological data, distribution data from about 5,000 herbarium records that I looked at and personally identified, and then ecological data from those records, so the elevation that a species occurs at. And then you can also calculate things like, how big of a distribution does this species have, or how broad of a niche does it have? And then to kind of get at some of these questions of the evolutionary history of this group, estimated diversification rates. I did that in BAM for the purpose of this talk. I've done it more recently in another package called RevBase. That's to estimate how fast speciation occurs. And then to correlate rates of diversification with species traits, carried out a number of phylogenetic generalized least squared regressions. So regressions essentially between speciation rates and species traits. And then to kind of try and get at the correlation between diversification rates in this group and rates of mountain uplift, used the analysis R-Panda. This is an R package that I think is really interesting, and looked for an association between these uplift rates, which were inferred from geological data, and speciation rates. Because that's not super intuitive, and I recognize that, I did provide a little bit of a cartoon here of an example of how this works. So if you were to imagine that evolutionary time was on the x-axis, going from left to right, from old to today, and you had two different things on the y-axis, some environmental variable, it can be elevation of the Andes, it could be carbon dioxide concentration or global temperature through time. You can see that that yellow environmental variable line goes down. So whatever it is goes down through time. And in red, we have the number of species in that lineage. And we see that that goes up through time. And so in this case, what R-Panda would do is, in a kind of sophisticated way, look for that association. And in this case, would demonstrate that there was a negative association between the environmental variable and that speciation in that lineage. The timeline of evolution of Phlegmariurus in the Neotropics is pretty interesting, I think. This is a group that, as a whole, again, about 150 species, is about 45 million years old. Now, it's pretty old, but again, keep in mind, that's only about 1/8 the age of the family as a whole. And you'll see by looking at this tree that most divergences in the genus occurred in the last 10 million years, which actually corresponds pretty well to the final uplift of the Andes Mountains. And I really want to draw your attention here in the bottom to the species that are indicated with that green bar where it says Crassus group. Those are the terrestrial species that we find in these alpine zones, the Phlegmariurus Crassus group. That whole clade, which includes about 75 species or so, 70 species, I lied, is about 7.5 million years old. So about half the diversity in this whole clade has just come up in the last 7.5 million years. So it's quite young. And that's what these species look like. I think they're just absolutely gorgeous plants, really interesting organisms, and phenomenally diverse. And as I suggested earlier, these are the species that tend to be very rare and have small ranges. So looking at the kind of relationship between elevation and diversity in the group, this is a really interesting history, again, where we have the Phlegmariurus crassus group at high elevations that is an extremely rapidly diversifying group. So if you look at this figure on the right, we have on the x-axis diversification rate, on the y-axis mean elevation that a group of species occurs at, and each of these circles represents a species group within Phlegmariurus in the Americas. And kind of the elephant in the room again, right, is this this big green circle, right? That is the Phlegmariurus crassus group. So you can see that that's a big group of species, rapidly speciating, and they're at high elevations. So that's kind of driving the remarkable diversity of Phlegmariurus in this group. So what about diversification in species traits? What's driving this rapid speciation in this clade? I've got a number of scatter plots here. These are these phylogenetic least squared regressions. There's a lot going on here, but I'm going to just walk you through it really quickly. Okay, so there's 4 plots, but each plot is quite similar. So each dot in these represents a species of Phlegmariurus in the American tropics. And the color of the dot corresponds to the species group that it belongs to. So you'll see those green dots, again, are the Phlegmariurus crassus group. On the y-axis of all these scatter plots is net diversification rate, so some estimate of how rapidly these taxa are speciating. And then on the x-axis, we have kind of 4 different traits that we can associate with these species. And the 2 I really want to draw your attention to are these ones in the red boxes. So at the top left, we show that we have a negative correlation between net diversification rate and species range size. So that's to say that the most rapidly diversifying lineages within Phlegmariurus are those with small ranges. And you can see that those tend to be the Phlegmariurus crassus group, no surprise there. And then down in the bottom right, we have a very strong positive relationship between elevation and diversification rate. So species that occur, again, at high elevations are the ones that are diversifying most rapidly. I don't think there's anything particularly surprising there, but it is nice to have some numbers kind of backing up this pattern. Now, to look at it in a slightly different way, I'm just going to talk through this figure real quick. It's a little bit complicated, but I'll walk through bit by bit, and I think it'll provide some interesting new insight into the evolution of this group. So the first thing I want you to notice is that we, again, have that same evolutionary tree of Neotropical Phlegmariurus in the background here that is time calibrated. And in red, we have the Phlegmariurus crassus group again, okay? And you'll see that that clade, again, is about 7.5 million years old. We actually include a little bit of a stem lineage there, so it's closer to 10. You'll see a dotted green line going from left to right and going up through that time. That is our best estimate, kind of a smoothed line based on geological data of the elevation of the tropical Andes through time. Right, so if you go all the way to the left, 45 million years ago, the tropical Andes basically didn't exist. And if you go all the way to the right today, the average elevation is almost 4,000 meters. I have a gray dashed line indicated there, and that shows that at about 10 million years ago, right when the Phlegmariurus crassus group kind of comes into being, we get to the point where the Andes probably started to break through tree line. Okay. And then the last couple things are these 4 lines that you see kind of diagonally going from bottom left to top right. And those lines are lineage through time plots. And what lineage through time plots show just that. They show the accumulation of lineages in a clade through time. Now, the black line, which is just a straight line, is kind of the line that we would expect if in the evolutionary history of Neotropical Phlegmariurus, these species accumulated new species at a perfectly constant rate. Okay, so that would be that there was never any variation in how fast new species were forming. The red and the blue lines are actual models that we ran that are kind of null models based on the same concept. Based in the concept that we would have just constant generation of species and that there would be no environmental input impacting rates of diversification in the group. And the yellow one is the real line that we see. Now, it's a little bit hard to interpret what's going on here, but I'll just tell you the main point is you will see that for much of the evolutionary history of Phlegmariurus, again, up to about 10 million years ago, that yellow line dramatically lags behind all of the expected models. That's to say that Phlegmariurus was accumulating species more slowly than we would expect based on the number of species that it has today and how old it is. And then just in the last few million years, it starts to accumulate new species at a very rapid pace, which is consistent with this relationship of diversification and uplift. Now, those R-panda analyses that I talked about before are related to this specifically, and how that works is it compares a bunch of different mathematical models, some of which include the height of the Andes through time as a predictive variable, and some of which don't. The ones that don't are the number 9 and number 10 on our kind of best ranked list here. So all 8 of the remaining models, which include the height of the Andes as a predictor of speciation rate in Phlegmariurus, outperform those models. So it really provides very strong support for the notion that there is a positive relationship between the change of the Andes through time and diversification in this clade. So did the rise of the Andes drive diversification in Neotropical Phlegmariurus? Yes, absolutely. We have a strong correlation of rates of uplift and diversification. And it's an interesting thing in which we see species at high elevations, and those with small ranges, are rapidly diversifying. And you can see where I'm going with this. These are rapidly diversifying species that end up stuck on the top of mountains with very small ranges. And this is a pattern that's driven by these Alpine taxa and the Phlegmariurus crassus group. So it's, again, these species that are growing in these sorts of habitats that are driving the exceptional diversity of this particular group of club mosses in the Andes. Again, remember that club mosses overall are not particularly diverse, but if you go to the northern Andes, they certainly are. But as I kind of touched on at the end of this, there's a really interesting component of this, and that's that, you know, the island life, the sky island life at least, coincides quite often with rarity. Like I mentioned previously, rapid speciation in Phlegmariurus tends to be associated with species that have very small ranges. And again, that's very commonly members of this Phlegmariurus crassus group. So for example, this species that I have a photo of here, this is a species that is only known from a single mountaintop in the department of Antioquia in kind of Western Colombia. And there's many species that fit that model. So it's a pretty good deal if you make new species frequently on the top of these mountains. But there's a little bit of a payoff, a little bit of a trade-off in that they tend to be very rare. And here's a little bit of a graphical representation of it. For those that have spent a lot of time in the tropics, this will come as no surprise, but for people who have not had the opportunity, and I highly recommend that you seek it out once this pandemic is over, the mountains of the Andes are a tremendous mountain range, but they're very patchy, right? And so if you see in white on this map are all the areas with elevations below 1,500 meters elevation. The green is all the area in northern South America and Central America between 1,500 meters and about 3,200 meters, which is the cutoff, again, for the lower limit of these alpine zones. And the blue areas are this alpine habitat at really high elevations. And as you can see here, there's a lot more continuity of lowland habitats, the white stuff, than mountainous habitats. And especially these alpine habitats are extremely fragmented naturally. Right? So you can imagine if you're a species that likes to live in lowland tropical forests, well, you've got a lot of opportunities to move around throughout the landscape. But if you have to live at really high elevations, it's pretty hard to make it happen. And I went and just kind of wanted to compare the distributions of some of these forest and alpine species. And the way I did this is I actually just made up a little code to randomly select some names. So this is actually really random. On the left are these epiphytic species that we find in the forest. And on the right are 3 terrestrial species that you find in alpine zones. And when we compare their distributions, we find a really interesting pattern that I think is characteristic of what you see in the genus as a whole. So looking at the forest species, we find that most of them have pretty wide ranges. So we have 3 different ones here with 3 different colors. There's kind of an orange, a red, and a yellow one. The dots that you see, there's several different shapes, and some are easier to see than others. Are actual specimen occurrences, and then I have kind of just coarsely modeled where these species occur. So you can see that a species like Phlegmariurus dichotomous, which is a species that's well known for growing in extremely low elevations, is tremendously widespread. And even other things that grow at somewhat higher elevations, like Phlegmariurus curvifolius or even Phlegmariurus tubulosus, are somewhat less widespread, but they still have pretty broad ranges, right? Even the least broadly distributed of these Phlegmariurus tubulosus covers all of Costa Rica. In contrast, if we look at some alpine species, we really see a different pattern here. Again, I have the same kind of information here, but I want you to pay really close attention to the dots on the map here, because I drew these polygons, and they're probably, to be fair, kind of considerable overestimates of how much habitat could actually be occupied by these species, because they cover habitats that's also too low. So you'll see that Phlegmariurus weber-baueri, mostly in Peru, has a kind of broad range, but it's very patchy. It really just occurs in kind of 3 areas, kind of south-central Peru, north-central Peru, and then on the Peru-Ecuador border. Phlegmariurus transida, a huge species that gets well over 1 meter tall, is only known from central Ecuador. Phlegmariurus venezuelanicus is only known from a few mountain ranges in Venezuela. So these species, in contrast, have really restricted ranges. I know you might be saying, all right, Wes, yeah, you didn't actually randomly select these. You picked really bad examples. And I actually didn't. If I wanted to pick really rare plants, I'd show you these. There are many species of Phlegmariurus that are local endemics, things that are only known from a single mountaintop or maybe a couple of mountaintops. And so I'm just showing a few examples here, but these are all species that are only known from one or a handful of localities. And this kind of seems to be the rule rather than the exception. So rarity is common in alpine club mosses in the American tropics. I went through and kind of counted up the numbers, and 1/4 of the species of Phlegmariurus in the American tropics that live in alpine zones are single mountain endemics. They're only known from one place. That's only about 1/10 of the species that grow in the forest that are kind of restricted to a single location. And the actual percentage of kind of single population endemics is actually probably higher. And I think it's the fact that we find the common species, right? There's probably a lot more really rare species that we just haven't found yet. And a really interesting thing, which you can kind of get a sense a little bit with from this photo, is that these species are actually often very locally abundant. So they're only found on one mountaintop. But rarity in this context really just refers to small ranges, and they're often quite common within that extremely restricted range where they occur. So it really boils down to, I think, this kind of diversity-rarity paradox. So islands, whether they be in the ocean or on top of mountains, promote speciation, right? They're isolated, small areas, limited gene flow between them, and low rates of migration into and out of them. On the other hand, islands promote extinction in some cases because they're isolated, they have small areas with limited gene flow and low rates of migration. So as I mentioned at the beginning of this talk, and I'm coming back to now, diversity and rarity often go hand in hand. And really, kind of fundamentally, alpine systems are unstable. We know that in the Andes, there have been dramatic climate shifts in alpine zonation, right? So just recently, since the early 1800s, the snow line on Mount Chimborazo in Ecuador, a very big mountain in Ecuador, has gone up 200 meters just in 200 years. And we know that much bigger shifts occurred repeatedly through the Pleistocene. Those obviously have huge impacts on species that grow on the tops of these mountains, right? If the alpine zone just disappears and you can only grow in the alpine zone, well, you're out of luck. So some of these species that are growing in these habitats are likely to just be pushed off the mountain in the future as habitat continues to get warmer. But in kind of the long term, this chaos really does offer opportunity. It's the same system that first afforded the opportunity for Phlegmariurus to diversify so successfully as these mountains grew up, and the subsequent kind of fluctuation of habitat availability continues to kind of erase everything and then let it start anew, which really drives these spectacular mountain biodiversity hotspots that we see in the Andes and other parts of the world. So kind of the tropical club moss takeaways that I hope you come away from this talk with is that club mosses are an ancient lineage, but they do have a lot of evidence of very recent diversification and ecological success. So I think you really can teach an old plant new tricks. Mountain uplift really does equal opportunity. Club mosses are one of the few lineages that really successfully capitalized on this. And so club mosses did a really great job kind of adapting to life on the ground again. People in temperate regions know that there are many terrestrial club mosses out there, but this lineage went into the trees and then came back down again and did very well. And alpine zones, I think, are inherently risky. They allow for rapid speciation, but they also have with them a high rate of extinction, and rarity is common. Coming up next, kind of future directions, ongoing things, working on this question and related ones with some bigger data, more species. So we now have a next generation sequencing technique where we've generated data for many species for about 450 genes instead of just the handful that I had a few years ago. I'm working on linking phylogenies with models of habitat connectivity, kind of the size of ranges across this biodiversity hotspot. Also interested in understanding the history of hybridization in club mosses. It seems like it's something that happens quite often, but seems poorly documented. So we're using some new phylogenomic toolkits to kind of get at that. And we're also really interested in integrating more fossils, and I'm working with some colleagues at trying to understand the evolutionary history of and relationships of a Jurassic club moss from Central Asia, actually. I'll do a quick pitch here in case anyone's interested. In December, I will be teaching an online, a 5-part short course on ferns and lycophytes of Central America through the Eagle Hill Institute. I think it's a really exciting opportunity, and given that a lot of us can't go anywhere this year, I think this might be an interesting opportunity for some folks to learn about Phlegmariurus and many other plants, looking at identification, diversity in these groups, biogeography, and ecology. And if you're interested in that, please go ahead to www.eaglehill.us. That'll be on weekends. And I have to thank a ton of people that made this work possible. Science is a collaborative effort, and I never would have been able to do this without the support of a lot of good friends and colleagues. I'd like to thank plant collectors, herbarium staff, those people don't get enough credit, and all the sort of research that I talked about today is reliant on people that do that work. I did my PhD in under David Barrington's direction at the now Barrington Sundew Lab at the University of Vermont. Thanks to Emily Sessa and the folks in the Sessa Lab at the University of Florida, who really had a great time working with them on lots of different projects there. I just finished up there now at the Antonelli Lab here in the University of Gothenburg, doing some new research. I'd like to thank folks at the Botanical Research Institute of Texas for all their support, especially Alejandro Vasco for inviting me to talk here today, and all these people and institutions, too many of whom to name. And at that point, I am very glad to take any questions that there might be. Thank you.
Wes. Thanks for a great. Oh, can you hear me? Yes.
Yes, I can hear you.
Thanks for a great talk. It was very interesting, and thanks to everybody who came again to this video. If you came late, you can just go back. Now we can go back in time and see the whole seminar in this link. There was a great talk. Thank you so much. And so we have some comments. I will encourage people to write their questions if they have, but I'm going to show some of the comments and maybe can talk about some of the things. So the first comment is from Manuela Del Forno, also a research botanist who was helping here behind the cameras. She said, fascinating, a very similar story, of course, in the genus Cora. Lichenized fungi in Basidiomycota. Can you see the comments, Wes, here?
I can see them. I have my phone out here. I'll put it here.
Oh, okay.
So I don't look down the whole time. Yes.
Perfect. So Cora, it's when you were talking about the super restricted distributions of them.
I've heard of that genus. I can't remember what they look like, but I have heard that it's a very diverse group.
Yeah. Okay, here we have a comment from Bianca Canestrado. Wes, there are several species of Phlegmariurus in the Atlantic rainforest of southern Brazil. You have to check them out here. So maybe you can talk a little bit about this connection.
Hi, Bianca. Thank you for watching. It's been quite a long time. And yes, I'd love to go to the Mata Atlantica and see these plants. They're super cool. So, yeah, thank you.
Okay, there is another comment here from our friend Yader. You have to come back to El Salvador and look again in Monte Cristo, now that we have some facilities to ask permission to collect.
Nice. Yes, I'll give people a little bit of background here. I think it was back in 2016, I went and visited Yader in El Salvador, had a wonderful time. They have this beautiful park in the northern part of the country on the border of Honduras and in Guatemala. And we saw some really just incredible Phlegmariurus, some species that I've seen nowhere else. And at the time, we did not have permission to collect them, unfortunately. But I would love to go back up there and learn more about those plants. And of course, would love to see you, Yader.
Great. Okay, I have here another comment of one of our research associates. His name is Harold Keller, and he says he wish he could talk to you because he has been at the University of Gothenburg and did research with his friend, Professor Uno Eli.
Oh, very cool.
So Harold works on, oh my God, Manu.
Plants.
No, fungi.
Fungi.
But I forgot. He's gonna kill me. Okay, so I we will leave people some time for questions. I do have a question for you, Wes. With this like super restricted distributions, what we are seeing in this group, do you know anything about the conservation status, or should we be like really worried about it.
Yeah, that's a really good point. We don't know for the most part. This is something that we have been talking a little bit with some folks at some different institutions. I've actually been in touch with people in Colombia, of course, as you know, and also some folks in Ecuador who are very interested in this. And there's some species that we do have IUCN red list assessments for, but that's only a minority of them. And of course, there's this very significant reality that even beyond having assessments of how rare species are, there's a very real problem with habitat degradation in these alpine habitats. They are increasingly, they get burned in some places quite often, and there's a lot of impact from agriculture in some cases, grazing. Those are certainly threats to these plants, I think. Although I would hazard a guess that for many of them, they are probably a little bit less threatened than some other species that grow in the same habitats. They seem to be somewhat resilient. The one thing that I do worry about with them quite a bit is water usage. So as paramos continue to be used as sources of water in some cases, if that changes the hydrology of these systems, I think that some Phlegmariurus species may be quite rare.
Okay, great. I had a couple of questions, but we are getting questions from the chat, so I want to put them. This is from our friend Michael Sandu. Wes, what is the story with Isoetes and Selaginella? Do they have similar stories in the Andes?
Yeah, that is a really good question, Michael. And I saw Carl Taylor was in this chat at one point, and he is, world's expert in Isoetes. So he would certainly know more than I do. Certainly, with the case of Isoetes, we see that Isoetes often occur at very high elevations in paramos, and they're generally aquatic plants, as you know. There's actually in this slide that's up here right now, which is from a paramo in Colombia, there is Isoetes in that little pool of water. And so in some case, I think they have even patchier distributions because, they can only occur in little ponds in these habitats. So I suspect that Isoetes would really show this pattern, and it's a group that desperately needs more study in the American tropics. Selaginella is an extremely diverse group, and there are a number of people doing really important work on that. But as you know, Selaginella tends to be a species at a bit lower elevations. And while I think the rise in the Andes has contributed to its rapid diversification in some contexts, I doubt that that is a group that has this sort of Sky Island system really playing in its history very much.
That's great. Look, Carl Taylor is here asking if there were Isoetes in that pond.
Yes, Carl. Carl, there was, so happy you're here, Carl. There are Isoetes in that pond, and I collected them. And there's, I think, a specimen at Britt. I collect them. If you saw a photo of an Isoetes early in the talk, there were 3 Isoetes on a rock. Those were 3 Isoetes from this pond right here, and they were put on one of the rocks in this photo, actually. So. Good question.
That's great. So we have another question here from Doug in Wales. You've talked about the sporophyte stage. Is anything known of the gametophyte stage?
Perfect question. Yeah, Eddie Watkins will want to hear about this, right? Yes, I only talked about the sporophyte stage, and you are correct in that I only told 1/2 of the story here. And just to give a little bit of background for people who might not know about these parts of the life cycle of ferns and lycophytes, both ferns and lycophytes have sporophytes and gametophytes that live independently from each other, right? So the gametophytes germinate from the spores, and then those later go through sexual reproduction and produce a new sporophyte. But the two are separate from each other. And the plants that I talked about, all these hanging plants and bright red plants, were the sporophytes. The gametophytes of Phlegmariurus are subterranean, so they actually don't do any photosynthesis. They rely on relationships with fungi for nutrients, actually. And we know very, very little about them. Their morphology has been described for some species, but not for most of them. And we know almost nothing about their ecology or about the reproductive history of these plants, which is really a shame. The reason why it's that way is simply that it turns out to be really hard to find these things in the field. But it's something that really warrants closer investigation, and I think could open up a whole lot of really interesting things about the ecology and the evolution of these plants. So yeah, it's a really good question.
I agree too. There's just a lot of things to learn about that other stage, and I think Doug and Wells also say this after this question in particular: are they still obligated mycorrhizal associates, or having dropped out of the trees, are they free of this stage? So I guess you touch a little bit.
Yeah, both the ones in the trees and the ones in the soil seem to be obligate mycorrhizal associates, which is quite interesting. But their morphology does change quite conspicuously, much like we see in gametophytes of ferns that switch between growing on the ground or growing in trees.
Great. And I think this is maybe the last question we're going to take. Yes. So it says, can you speak briefly on the ecological relationships of lycophytes in alpine environments? And could any of these relationships correlate with rapid diversification? This is Lauren Bansbach.
Okay. Hi, Lauren. Thank you for that question. Yes, there's a lot of really interesting things that are going on here. And the question that you asked touches on a really important theme in the evolution of these plants. The reason, in part, why species that radiated into these alpine habitats in the tropics were able to speciate so rapidly, in part, is that these were totally novel habitats. So there was really nothing there growing because they were brand new. But many lineages were unable, for one reason or another, to successfully transition to them, right? You can imagine if you're growing in a tropical forest and suddenly an alpine habitat opens up, you need to make a lot of ecological shifts to be able to do that. And most tropical plant lineages couldn't do it. So the two options were either figure out how to transition into this new habitat and survive, or disperse from some temperate place that might be ecologically more similar. If you look in this photo, actually, you can see these grass-like plants that are drooping over the edge of the water. Those are a kind of sedge, and I believe that they belong to a lineage that has successfully dispersed from temperate regions. But it turns out that most of the things that you would see in this photo, their closest relatives are tropical. And Phlegmariurus is one of these very few that was able to successfully transition in that way. How they were able to do it is hard to say. One thing that is a big constraint on many species is the amount of sunlight that they get, and UV radiation in particular. Seems like Phlegmariurus gets around that by becoming red. And the other component might be living in rather nutrient-poor soils in some cases, and waterlogged soils. And that could simply be a holdover from a very old history, that there's a lot of lycophytes that grow and have grown probably for many, many millions of years in nutrient-poor and waterlogged soils. And Phlegmariurus retained some of those traits, which might allow them to have successfully transitioned here. And I think that probably did in part drive the very rapid diversification of this group.
Great, thanks so much, Wes, for all your talk, for all these answers. I was going to share here. This is Wes's website. So if you want to get in touch with Wes or know about what he's doing or learn a little bit more about what he has told us today, please go there. As we can see, there are a lot of things to still learn and explore, right, Wes? in
Yes.
the tropics and other and lycophytes. So thanks so much, Wes, and thanks to everybody who stayed and made the change with us with this new link. Again, if you came late to the lecture, you can just go back, and this video is going to be available in the BRID YouTube channel. So with this, I would like to also put this is our next lecture. So there's gonna be information on the link of this lecture here in BRID.org lecture series, but please come and join us for in December 1st for this Textiles on Trees, Uganda Bark Cloth from Leslie Robertson. So thanks so much, Wes, again.
Great. Thank you, everybody.
Thanks, everybody, for joining us today. Bye.
Okay. Bye.